All right, so to start off, I just want to review chemical reactions. This slide should look familiar. Yes? Like we have seen this slide before? Good. Okay, so I just want to review. A chemical reaction is when atoms or groups of atoms and compounds, they're going to rearrange and form different substances. And I just want to start with our generic formula for a chemical reaction. So we put reactants on the left. Then we have that arrow that means yields or it produces, and products go on the right. All right, so today we're going to talk about enzymes, and enzymes are going to help speed up chemical reactions. So I want to start with just some chemical reaction vocabulary, okay? So there are lots of different types of chemical reactions. Two that we're going to pay attention to that enzymes participate in are called synthesis and decomposition reactions. Okay. So the first one is a synthesis reaction. These are reactions where you build something from smaller pieces. So I'm just going to give you an example of what a synthesis reaction might look like. So maybe we have element A plus element B. They're going to combine and make compound AB. The next type of reaction is called decomposition. Decomposition has the word decompose in it. What does it mean if something decomposes?
Breaks down over a small period of time.
It's going to break down. So then let's say we have a larger compound, let's say like compound AB. It can decompose into element A and element B. So now we can rewrite our generic formula for a chemical reaction. So we still have reactants and products, but now I'm going to add. Our reactants, when we're looking at enzymatic reactions, are called substrates. Okay. So substrates, which are reactants, they're still going to make a product, but in order to do that, you need an enzyme. So above the arrow, do you see how I have the word enzyme? Okay. It has to go above the arrow. It means that the enzyme participates in the reaction, but it's not a reactant and it's not a product. Okay, so it's just kind of there. And so finally, that brings us to our definition. What is an enzyme? An enzyme is a protein, and it just speeds up reactions. Anything that speeds up a reaction is called a catalyst. Think back to Monday this week when we did the McMush lab. Do you remember one of your tests you had to have the simple sugar test, you had to take your test tube and put it in the hot water? Okay, that's because you need the hot water just to speed up that reaction. So that is like an example of a catalyst. All right, here's just a little bit more vocab. This is pretty similar to our first slide. So if you don't need to add vocab, you don't have to. But just our starting materials are our reactants. When we're talking about enzymes and reactions, they're also called substrates. And then our products is whatever is produced in that chemical reaction. All right, so let's talk about chemical reactions and energy needed. So you don't have to write anything on this slide, but do take a look at the picture up here. So every single chemical reaction needs some sort of energy to make it happen. Okay, we call that energy activation energy, and think like you need so much energy to activate that chemical reaction. Okay, so here's just like a graph. For this reaction to happen, you have to have whatever the measurement would be. So much energy. Think about a burning candle. Okay, when you burn a candle, a chemical reaction happens where you take the elements in the candle wax and it ends up making like carbon dioxide and water vapor. Now, do candles just spontaneously light? No, right? You have to have so much energy put into your candle before it can actually burn. Okay. Same is true for all chemical reactions. So how catalysts work is they just speed up chemical reactions by lowering that activation energy. So if you take a look at the graph up here, this at like the very top of this blue line, that's how much energy the reaction typically needs without the catalyst. Okay. When you use a catalyst, do you see how it just lowers that activation energy? So that's how we end up speeding up chemical reactions. So do go ahead and copy the bold words down in your notes for this slide. So that brings us back to enzymes. So enzymes are just biological catalysts. So they help speed up reactions that happen in living things like you. They are essential to life. Okay, so they help reactions happen in a more reasonable time frame. Like when you digest food, you have lots of enzymes in your digestive system that help you break down your food, and you're able to turn it into usable energy in like a time frame that's actually possible that you can actually use that energy. All right, when we name enzymes end with the three letters ASE or ACE. Okay. And the name of the enzyme, it correlates to whatever substrate they work on. So for example, lactase is going to break down lactose. Sucrase is going to break down sucrose. Okay. So the name of the enzyme, it tells you what it's going to react with. And then what three letters do you look for to identify an enzyme? What three letters will they end in? ASE. So you're looking for ACE as the ending. That's how you know it's going to be an enzyme. All right, so let me show you an example of what an enzyme reaction might look like. Okay, so here's an example of lipid digestion. So your body would use the enzyme lipase, and it's going to help break down lipids into its building blocks, or just the monomer. So the glycerol and the fatty acid. Okay. So the enzyme in this reaction is lipase. Okay. The substrate or the reaction or reactants are lipids, and then the product is going to be your fatty acid and glycerol. So then if we were just to put this in our generic formula, our reactants are going to be our lipids. Over the arrow, I just put the enzyme lipase, and then products, fatty acids, and glycerol. Okay. So can you see how all I did was just take these three items up here, and I just put it into like our generic formula of a chemical reaction? All right, take a look at this picture up here. Which enzyme do you think is best structured to break down the lipid? Okay, so here's a lipid up here. So which option do you think would best break it down? Option 1, option 2, or option 3, throw a number up there. Nothing happens if you're wrong, so put a number up there. Which enzyme do you think is best structured? We're looking at the structure, though. Which structure looks like it would best break down the lipid over here?
Lipid?
Yeah, the lipid is the picture in the top right. Okay. Why 2?
Because each one, the like things fit in.
Okay, it is option 2. So the shape of the enzyme has to match the shape of the substrate. Do you see how like option 2, it looks like it fits in there?
Yeah.
Okay, a good example is think of like a lock and a key, right? In order for my key to unlock the lock of my door, it has to be the right shape, yes?
Yeah.
Okay, so another analogy to think of enzymes and how they work. Who's a great quarterback these days?
Amen.
Lincoln, raise your hand. Our C-team
Let's just
quarterback
go
right
with
there.
Lincoln. Lincoln is pretty good at throwing, okay? So Lincoln can go out in the hall and he can throw the key at my door, yes? Right? Statistically speaking, he could eventually throw my key in the door lock because it would fit, yes? Now, would it maybe take him a lot of throws, like a million, 10 million, 100 million? Who knows? Statistically speaking, though, like if he did it enough, could he like throw my key in the lock?
Yeah.
Yes. So think of an enzyme as like hands right by the door. It's going to catch it and put it in. Is that going to speed up the amount of time it's going to take Lincoln to actually get my key in the lock?
Yeah.
Yes. That is what enzymes do in chemical reactions in our body. Maybe. Okay, so go ahead and just take a look at the diagram up here. Okay, so this goofy looking green thing, that's our enzyme. Okay, do you see how the enzyme has a very specific shape?
Yeah.
Okay, so it has to bind to a substrate that matches that shape. Okay, so here's the substrate in orange. Once those two bind together, the chemical reaction starts, and you end up turning that substrate into products. So do you see how that orange turned into purple and yellow? All right, once the chemical reaction's complete, the enzyme and the products separate, and then the products, they're done, right? Whatever your reaction is, it's made. And let's say it's the digestion one, your glycerols and fatty acids, they're released. Your enzyme remains intact, and it's ready for another chemical reaction. So your enzymes are reusable unless they become denatured. Okay, so enzymes can become denatured, which means they become deformed. Okay, so take a look at this picture down here. The one on the right, it is a denatured enzyme. Can you see like the shape is no longer intact? Okay, so like if I were to change the lock on my door, is my key gonna work anymore?
No.
No, same thing for enzymes. If their shape changes, the substrates no longer can bind to them, which means they no longer work. They're no longer going to speed up that chemical reaction. And what causes an enzyme to denature is typically if you have a change in your temperature or a change in your pH.